Sulforaphane and its effects on cancer and disease. What exactly is sulforaphane? It's a compound found in certain vegetables that's packed with health benefits. It's made from a substance called glucoraphanin. When vegetables are chopped or chewed, thanks to an enzyme called myrosinase, this process turns glucoraphanin into sulforaphane, which is a special sulfur part in its chemical makeup, making it great at boosting our body's defense systems. It's especially good at fighting off damage from pollution and stress on a cellular level, helping to keep our cells healthy. You'll find sulforaphane in cruciferous vegetables like broccoli, kale, Brussels sprouts, and cauliflower. But broccoli sprouts are the real stars, loaded with the stuff. The amount of sulforaphane these plants can offer depends on several things: the type of vegetable, how it was grown, and the enzyme myrosinase that activates sulforaphane. Young plants and sprouts usually have more of it. Factors like the soil they're grown in and how much sunlight they get and whether they've had to fend off bugs can also affect how much sulforaphane we can get from them. This is one reason why eating a variety of these vegetables is often linked to good health benefits, as the sulforaphane content can vary widely. After ingestion, sulforaphane is quickly taken up by the intestines and transported to the liver, where it undergoes a sophisticated detoxification process. This mercapturic acid pathway is essential for converting sulforaphane into forms that the body can either use for protective mechanisms or easily excrete. The efficiency of this process is crucial for sulforaphane's protective effects against oxidative stress and inflammation. Variability in this metabolic pathway among individuals highlights the need for personalized nutritional approaches to maximize the health benefits of sulforaphane. The conversion efficiency of glucoraphanin to sulforaphane, which is critical for its bioavailability, is highly dependent on the presence of the myrosinase enzyme. And this enzyme's activity can be affected by several factors, including the method of food preparation, not surprisingly, the individual's gut microbiome composition. Furthermore, the health of the gastrointestinal system plays a pivotal role, as it can significantly influence the enzyme's activity and consequently the amount of sulforaphane available for absorption. This underscores the complexity of nutrient absorption and the interplay between diet, gut health, and genetics. The form in which sulforaphane is consumed, whether as free sulforaphane, its glucosinolate precursor, or in a synthesized form, affects its bioavailability and the body's ability to utilize it. Supplements may provide sulforaphane in a more bioavailable form, but the presence of active myrosinase in natural sources is critical for the conversion process. The interaction between dietary sources of sulforaphane and the gut microbiota further complicates its bioavailability, highlighting the importance of considering the form of sulforaphane when assessing its potential health benefits. In this study from a short-term crossover clinical trial in China, the study investigated the bioavailability of sulforaphane from two types of broccoli sprout-derived beverages, glucoraphanin-rich and sulforaphane-rich, so GRR and SFR. The research revealed that sulforaphane's bioavailability was significantly higher when consumed in the SFR form compared to GRR, with urinary excretion rates of sulforaphane and its metabolites being about 70% for SFR and only 5% for GRR. This substantial difference underscores the importance of the form in which sulforaphane is ingested for maximizing its health benefits. Sulforaphane strategically targets and activates the NRF2 pathway, which enhances the body's arsenal against oxidative stress, a key factor in cellular aging and damage. It deactivates the NF-kB pathway, reducing inflammation and its role in promoting tumor growth and proliferation. This dual mechanism not only helps in fortifying cellular defenses against environmental stressors, but also plays a critical role in preventing the initiation and progression of cancer by modulating processes vital to cell survival and death. Sulforaphane elevates the body's detoxification processes, by turning on genes that produce phase 2 detoxification enzymes, aiding in the rapid clearance of toxins and carcinogens. Through the inhibition of histone deacetylases, it influences the epigenetic landscape of cells, potentially activating tumor suppressor genes and deactivation of genes that promote cancer growth. This influence on gene expression extends beyond detoxification, impacting cellular repair processes and the immune response, thereby offering a broad spectrum of protection against cancer and other diseases. By promoting the production of antioxidant enzymes, sulforaphane directly combats oxidative stress, reducing the risk of cellular mutations and chronic disease development. It effectively modulates the immune system's response to reduced chronic inflammation, a known precursor to many diseases, including cancer, cardiovascular diseases, and neurodegenerative disorders. The compound's ability to both directly neutralize harmful free radicals and influence the body's inflammatory processes underscores its potential as a powerful natural agent in the prevention and management of chronic diseases. In this study, published in the Journal of Antioxidants, it provides evidence that sulforaphane significantly impacts oxidative stress by activating the NRF2 pathway, which leads to the upregulation of antioxidant defense mechanisms within cells. Through clinical and experimental research, the review confirms sulforaphane's efficacy in enhancing the cellular antioxidant capacity, highlighting its potential to mitigate oxidative damage and its implications for chronic disease prevention. This detailed analysis demonstrates sulforaphane's role in modulating cellular responses to oxidative stress, emphasizing its value in dietary interventions aimed at reducing the risk of oxidative stress-related conditions. Sulforaphane operates as a powerful antioxidant, attacking and neutralizing dangerous free radicals that can damage cells, while simultaneously enhancing the body's own antioxidant defenses through mechanisms like the activation of the NRF2 pathway. This pathway's activation leads to a significant increase in the production of enzymes crucial for detoxifying harmful substances and protecting against oxidative stress, such as glutathione S transferase and the quinone reductases. These enzymes are essential for maintaining cellular health by preventing the accumulation of harmful oxidative products. I'm interrupting this video because I would love for you to grab my intermittent fasting quick start guide. You can click the link below and grab it now. Unlike many antioxidants that offer either direct neutralization of free radicals or support for the body's antioxidant defenses, sulforaphane excels in both arenas. It uniquely provides direct scavenging of harmful free radicals while also stimulating the body's production of phase 2 detoxification enzymes. This multifaceted approach allows sulforaphane to deliver a more comprehensive protection against oxidative stress, making it an exceptionally potent molecule for safeguarding cellular integrity against oxidative damage. The antioxidant qualities of sulforaphane are key in the fight against cellular damage that leads to chronic diseases such as cancer, cardiovascular diseases, and neurodegenerative disorders. By reducing oxidative stress, sulforaphane helps to preserve cellular integrity, supporting overall health and reducing the risk of disease development. This underscores the importance of including sulforaphane-rich foods in the diet as a proactive measure for disease prevention and health maintenance, highlighting its critical role in supporting the body's natural defense systems. This review from Oxidative Medicine and Cellular Longevity delves into sulforaphane's capacity to combat oxidative stress, emphasizing its unique ability to trigger the NRF2 signaling pathway, which leads to an increased expression of antioxidant proteins and enzymes, crucial for cellular defense mechanisms. Sulforaphane's effectiveness is partially due to its nutrigenomic effects, allowing it to modulate gene expression related to oxidative stress, thereby bolstering the body's innate antioxidant systems. This process underlies its potential therapeutic role in preventing and managing oxidative stress-induced conditions. Sulforaphane targets cancer cells by halting their growth cycle at various stages, effectively preventing them from proliferating. This action disrupts the normal progression of the cell cycle, a crucial process for cancer cell multiplication and tumor growth. The ability to induce cell cycle arrest makes sulforaphane a potent compound in cancer prevention and treatment strategies, offering a means to curb the spread of malignant cells. By triggering apoptosis or programmed cell death, sulforaphane ensures the removal of damaged or cancerous cells in a controlled manner. It influences vital apoptosis-related pathways and proteins, including capases and the BCL2 protein family, which are instrumental in the cell death process. This selective targeting spares healthy cells while effectively eliminating cancerous ones, highlighting sulforaphane's potential as a targeted cancer therapy. Sulforaphane also has an ability to inhibit angiogenesis, which is the formation of new blood vessels, and strikes at the heart of tumor growth and metastasis by cutting off the essential supply of nutrients and oxygen to cancer cells. This not only restricts tumor expansion, but also limits the potential for cancer cells to spread to other parts of the body. The anti-angiogenic properties of sulforaphane add another layer to its anti-cancer effects, making it a valuable component in the fight against cancer. In this study looking at sulforaphane as an emergent anti-cancer stem cell agent, it elucidates sulforaphane's potent anti-cancer properties, notably against cancer stem cells, by altering self-renewal pathways and modulating abnormally activated signaling pathways in these cells. Additionally, sulforaphane's inhibition of histone deacetylase enzymes plays a critical role in activating tumor suppressor genes, offering a multifaceted approach to cancer prevention and treatment. This highlights its potential as a complementary strategy alongside conventional anti-cancer drugs, underscoring its significance in developing future cancer therapies. Lastly, sulforaphane significantly enhances the body's detoxification pathways by stimulating the expression of phase 2 detoxifying enzymes. These enzymes play a critical role in transforming carcinogens, toxins, and other harmful agents into less toxic forms that are more easily excreted from the body. This enzymatic activity not only reduces the risk of developing cancer and other diseases, but also supports the body's overall ability to manage and neutralize toxic substances, reinforcing our natural defenses against environmental and dietary pollutants. Stay tuned for my next video looking at vitamin D and the aging process.